Good morning, everyone. Today I'd like to discuss Corynebacterium and its impact on our world. We'll explore the various requirements of Corynebacterium and its presence in nature, from habitat to morphology to biochemistry, as well as its host range, its antibiotic resistance and epidemiological and pathogenic properties. Corynebacterium is a diverse group of gram-positive, non-spore-forming, aerobic to facultatively anaerobic bacteria. Found in many different environments, they may cause opportunistic or even pathogenic infections in humans and animals. Given its importance, an extensive body of research has developed to explore its biology and biochemistry. Corynebacteria are present in a variety of environments, including soil, water, plants, and the microbiota of animals and humans. Visually, they are rod-shaped, measuring between 1 and 10 micrometers in length and 0.5 to 1.0 micrometers in width, displaying a distinctive bulged shape. Corynebacterium are gram-positive bacterial organisms, typically rod-shaped with a distinctive bulging appearance. Measuring between 1 and 10 microns in length and 0.5 to 1.0 microns in width. Corynebacteria are a fascinating species of bacteria with many cultural characteristics. Slow-growing and requiring complex media for optimal growth, colonies formed on solid media often resemble smooth, opaque, or translucent formations, offering insight into their delicate and unique biology. Corynebacteria are found in a variety of environments, from soil to water, and possess unique biochemical characteristics. These bacteria are characterized by their production of the enzyme catalase, but unlike most bacteria, they are unable to produce the enzyme oxidase or form spores. Corynebacteria are capable of producing a variety of metabolic byproducts, including acids, alcohols, and even gases. Genus Corynebacterium encompasses a broad range of species that can infect humans, animals, and plants. While some species may be benign and even provide positive effects, others can be detrimental and cause serious infections in those with weakened immune systems. Hence, it is essential to be aware of the various types of Corynebacteria, their natural roles, and how to avoid them from causing damage. Corynebacterium is a group of bacteria that inhabit a wide range of environments, including human body surfaces and our environment. Characteristic of this bacteria is the variety of virulence factors they produce, including toxins, proteins that allow them to evade our body's immune systems, and adhesins, which help them stick to surfaces. To better understand their growth and virulence factors, this research examines the various strains of Corynebacterium. Corynebacterium are gram-positive cells that play a role in the usual microbiota found in the human body, but can also become opportunistic pathogens. To combat their spread, it is essential to understand how Corynebacteria develop antibiotic resistance, which can involve efflux pumps, enzymatic modification, and target site mutations. Corynebacteria are a diverse and widespread group of bacteria, commonly found in the environment. Capable of transmission between hosts through contact, aerosolization, and contaminated food and water, their spread and potential for causing disease should be understood. Precautions can be taken to limit the risk of infection. Corynebacterium can cause a range of serious diseases in both humans and animals. These infections can vary from skin and soft tissue issues to respiratory and even systemic issues. Therefore, it is essential to be aware of the potential risks these illnesses pose and to take appropriate preventative action to protect ourselves and our animals. Corynebacterium is a pathogen commonly found in the environment which can cause infection in humans. The body's defense against it involves both innate and adaptive immune responses, of which antibody-mediated immunity is the most important in providing protection from recurring infection. Corynebacterium infections can be tricky to diagnose, as they are all too often not identified correctly. Laboratory testing of clinical specimens, like blood, tissue, or swab samples from the affected areas, is necessary to identify these infections. Species differentiation might demand certain testing methods, like DNA sequencing. Accurate diagnosis of these infections is essential in order to ensure the most successful treatment. Prevention of Corynebacterium infections is essential to combat its transmission. Vaccination, hand hygiene, and judicious use of antibiotics are common methods that can be employed to counter this kind of infection. By taking the required measures, we can contribute to safeguarding ourselves, our family, and our society. Corynebacterium infections can present a challenge when it comes to treatment, often requiring a mixed approach of antibiotic therapy and surgical intervention. The specific treatment plan depends on the kind of infection and should be tailored to the individual patient's specific condition, taking the severity of the infection into account. In some cases, a mixture of antibiotics and other medications may be necessary to adequately treat the infection. It is important to accurately diagnose and treat Corynebacterium infections correctly in order to control them. Several methods have been developed to do this, including examining a sample of the organism and using tests such as PCR. Gaining a deeper knowledge of Corynebacterium can help us successfully treat infections it may cause. Thank you.